This is Metabolism and Pancreas, an introduction, parts 1 and 2. In this video, you'll learn the functions of two important metabolic hormones, insulin and glucagon, and how each of those hormones are going to be antagonistic to the other. You'll be able to describe what happens after you eat a meal, what happens to the nutrients during gastrointestinal absorption. You'll be able to also know the endocrine and metabolic responses during post-absorptive states and what happens during fasting in terms of hormonal control. You'll learn as well the endocrine and metabolic responses to exercise and why this is important for us as clinicians. And finally, you'll be able to explain the changes in the metabolism for some of our patients that suffer from diabetes. Before we talk about insulin, glucagon, and all these other physiological processes modulated mostly by the pancreas and other organs, we're going to talk about four important nutrients. Amino acids, which are the building blocks of protein. Glucose, which is going to be the center or the star of the discussion. And we already know that when we have excessive glucose, we can store it in the form of glycogen in both the liver and muscle tissue. We also have glycerol and fatty acids, which combined make up most of the fat that's deposited subcutaneously. Now, take a look at this glucose molecule here. The breakdown or catabolism of this molecule happens during first a process called glycolysis. It generates a little bit of ATP, and this process happens in the cytosol of cells. It's independent of oxygen presence. The end product of glycolysis is called pyruvate, and pyruvate is further degenerated to become another compound called acetyl coenzyme A. We get acetyl coenzyme A from both pyruvate catabolism and fatty acid catabolism. So two different routes to get to this intermediary. This chemical compound enters another cycle called the Krebs cycle. In the Krebs cycle, more ATP is generated, some carbon dioxide is released, and one of its intermediaries will yet enter a third process that occurs in the membranes of mitochondria. So we need oxygen for this process to happen, and this process is called oxidative phosphorylation. Most of the ATP that our cells produce come from that process. The release of that process is water molecules. Now, what I wanted to point out on this slide is that there is a relationship between proteins, sugars or carbohydrates, and fat. The purpose of this slide is to illustrate the point that if our bodies are depleted of one of these nutrients on top, there are ways in which we can bypass some of these cellular processes in order to obtain, synthesize, and get more of whatever it is that's depleted. So this is a picture of the pancreas. The pancreas is both an exocrine and an endocrine gland, and we can see here the pancreatic duct, which composes or contributes to that exocrine part of the gland. We know that there's a couple of papilla or smaller nipples that drain into the duodenum, especially the second portion, and bicarbonate and other enzymes will be secreted there in order to assist with digestion. But again, that's the exocrine part of the gland. The endocrine gland part of it is modulated by this tissue here, which I'll show you in the next slide. All these are ducts that correspond to the exocrine part of the pancreas. What we're interested in is this area in the center. This is called an islet of Langerhans, and there's three types of cells there. The majority of them are beta cells, the pink colored ones. Then we also have alpha cells, which are colored in purple, and finally we have delta cells, which compose a minority of the islets. Beta cells synthesize insulin, alpha cells synthesize glucagon, and those two are antagonistic with one another. Delta cell synthesize another hormone called somatostatin, and this hormone in particular inhibits both glucagon and insulin. Notice here the location of neighboring adjacent blood vessels. Again, not surprisingly, considering that these cells are secreting their hormones into the bloodstream. This is a histological slide of the islet of Langerhans, but in this diagram you can actually see their organization. Most of the B cells are going to be centrally located, and most of the alpha cells are going to be in the periphery. So the perfusion cycle is from central to periphery. So if we recall that beta cells produce insulin, as they move from central to peripheral, that should have an effect on alpha cells producing glucagon. So that's how we know, or that explains why insulin, when secreted, is inhibiting glucagon. We also should know that the islets of Langerhans not only are sensitive to hormonal changes in nutrients like glucose, but also they are under the influence of the autonomic nervous system. Plasma glucose levels, or blood sugar levels. We know that normal fasting blood glucose should be within the range of 70 to 100 milligrams per deciliters. And we expect that insulin levels should be low at that concentration. You would see difference in ranges depending on which resources you read. For example, the American Diabetes Association provides another range. It's very close to the one I just gave you, but theirs is from 79 to 110. Know this though, clinically speaking, 126 milligrams per deciliter or above on 2 separate occasions is enough to diagnose diabetes mellitus. Hyperglycemia doesn't mean you're diabetic though. A lot of times we can become temporarily hyperglycemic, especially in periods of stress. So how do we regulate glucose in blood? Well, obviously by eating carbohydrate-rich products in our diet. That should last for about 2 hours after we have ingested them. We also reabsorb in the small intestine glucose through a sodium glucose co-transporter. Now, after 2 hours, then we rely mostly on hepatic glucose that has been stored there in the form of glycogen. So in order to get to that glucose, we need to undergo a process of glycogenolysis. And we can also obtain glucose from non-carbohydrate sources, such as gluconeogenesis. These processes happen in the liver. So we know that if we eat carbohydrate-rich foods, we're going to get glucose somehow into our bloodstream. But how does it get to the cells? Well, we need insulin for the uptake of about 40% of the glucose that's ingested. The rest is insulin independent. There are various glucose transporter mechanisms. The one that responds to insulin is GLUT4. And it does that by mediating a particular process known as translocation. Translocation means that the receptor is somewhere in the cell, and at one point it is expressed on the cell membrane. This happens mostly in muscle and fat, and 80% of the glucose we get from that's insulin dependent goes to muscle. Studies have shown that exercise improves this process independently of the concentrations of insulin, which obviously has a big impact in the way we work. There's, again, insulin-independent glucose uptake, and the receptors or the translocators that are active in this process that do not respond or are sensitive to insulin are GLUTs 1, 2, and 3. So this process happens by facilitated diffusion. When glucose is high in blood, that promotes the release of insulin from B cells in the pancreas. Now, insulin triggers this cellular process in which we see an acceleration of translocation of GLUT4 receptors for glucose. Again, these receptors were in the cell or within the cell, and in the presence of insulin, this activates the expression of those GLUT4 receptors on the cell membrane. Now, these are all examples of GLUT4 receptors receiving and facilitating the uptake of glucose. When insulin is in low concentrations, we refer to that as a basal state. where the intake or uptake of glucose is minimal. Now let's see what happens in the presence of an autoantigen known as GAD65 and how this plays an important role in the insulin-dependent diabetes mellitus or diabetes type 1. First, we have here an islet of Langerhans, and notice here that these cells look healthy, but the yellow ones are not, and they will die eventually. When these cells die and lyse or break, they release an autoantigen represented by these red dots. These are molecules of GAD65. Now, we're in the pancreas. This is pancreatic tissue. GAD65 is going to make it eventually in a lymph node that drains the pancreas itself. And eventually, there's going to be an antigen presenting cell that will engulf this autoantigen and present it on the cell surface of the same cell. When that happens, that attracts a T cell that's very aggressive, labeled here as an autoreactive T cell. These aggressive T cells will eventually migrate to the pancreas where they will then start attacking healthy B cells. And that's why GAD65 mediates this autoimmune reaction. Glucosuria is the term that we use for glucose in urine. We know that there is a threshold for glucose reabsorption in the nephron, and sometimes when it exceeds the glomerular filtration, this is what happens, glucosuria. We also know that polydipsia and polyuria are two symptoms that are very common in diabetes, especially diabetes type 1. Sometimes diabetic patients, IDDM patients, will be testing their urine for the presence of another family of compounds known as ketones. Ketones are a byproduct that happens or is generated when muscle and fat tissues are used for energy instead of glucose. The process in which insulin is secreted is actually very straightforward. Glucose goes up in B cells themselves, and that causes a membrane depolarization within that B cell. Calcium then goes intracellularly within those cells, and that facilitates the exocytosis of the secretory granules containing insulin. As we said before, acute hyperglycemia, or whenever we have high concentration of glucose in blood, that will stimulate or enhance insulin release. Also, the presence of certain amino acids. Some of the inhibitors, hypoglycemia, elevated free fatty acids, somatostatin, and catecholamines like norepinephrine. So physiologically speaking, this is what insulin does. In the liver, it inhibits glucose output and it promotes its storage. So think about it as conservation of glucose or storage for future use. It also promotes free fatty acid synthesis. In muscle, it increases glucose uptake. In fat, it does the same thing. It also decreases fat breakdown. It stimulates amino acid uptake, and we knew that it inhibited glucagon. If you think about it, this insulin hormone is actually very, very anabolic. It's all about creating and not breaking down. A lot of things can go wrong with insulin concentration. We're going to focus our discussion on the first two, type 1 and type 2 diabetes mellitus. Type 1, again, being the insulin-dependent one. In type 1, the majority of those patients you're going to see are young, or they have been diagnosed at a young age. You get type 1 diagnoses when about 70% of the B cells have been destroyed. We say that this is an absolute insulin deficiency, and we knew or learned earlier in this video that this is a T cell mediated autoimmune reaction, reacting to a particular autoantigen, GAD65. We also know that type 1 diabetes carries long-term adverse effects, and we're going to see some of them in peripheral tissues. Type 2 is non-insulin dependent diabetes, and the majority of diabetic patients correspond or are assigned to this category. This is a middle-aged typical individual that's being diagnosed, and it usually goes hand in hand with obesity and overweight. We know that in type 1, it was an absolute insulin deficiency. Type 2 is a relative deficiency of insulin. And a lot of times it has to do with something known as insulin resistance, which is the deficiency of target tissues responding to what insulin does, which is facilitating the uptake of glucose. Most patients, interesting enough, are hyperinsulinemic, meaning they demonstrate or can exhibit in a blood test high concentrations of insulin. Secondary diabetes is a little different. You get overproduction of insulin antagonist hormones, and probably one of the common ones are cortisol and catecholamines like epinephrine, but there are others. Gestational diabetes mellitus, or GDM, is very, very similar to the secondary diabetes. Again, it's an overproduction of insulin antagonistic hormones. A lot of times, women that suffer from GDM will eventually become diabetic type 2. There's also very few cases of hyperinsulinemia that result as the product of a tumor that is insulin secreting, but this is very, very rare. There's also something called insulin shock, which we will discuss in class in more detail. Although we have focused on insulin, there's also another hormone, glucagon, which again, we knew that it inhibited insulin. We know that it's synthesized in alpha cells, like we said earlier in the video. The major antagonist is insulin. The main stimulus is a drop in blood sugar. So I call this the hormone of hunger. Other stimuli, a couple of different amino acids. It can also be stimulated directly by autonomic innervation. And again, it's secreted during fasting, starvation, and if you are type 1 diabetic. It's inhibited by insulin and GABA. We will finish this video by listing the physiological effects of glucagon. It is definitely an energy mobilizer, whereas insulin did the opposite, all about anabolism and storing nutrients. Glucagon is going to do the opposite. It's going to promote catabolism. It has absolutely no effect on skeletal muscle, which is very different from what we saw in insulin. However, it acts on other tissues. In the liver, it promotes glycogenolysis and gluconeogenesis. So glycogenolysis, which is the opposite of what insulin does. It acts on adipose tissue as well by stimulating lipolysis, breaking down of fats. And pathologically speaking, there could be glucagenomas that are focusing on a high concentration of alpha cells, but those are extremely rare.